Go.
Okay, this is going to be part one of the Introduction to Microscope Lab. I will kind of walk you through how these microscopes work. My wife is running the camera, and there'll be times I'll be talking to her to come in and go out. You'll figure that one out. But this is a video microscope, and so it can project a picture so that you can see what I'm seeing. It's a little different from the microscopes we use in our actual lab. The controls are all the same, but it's set up just a trifle different. So if you do wind up working with a microscope, it's good to know that each microscope, the basic controls are always the same, but sometimes you'll go, well, how do I move the slide? How do I open the iris diaphragm? That may vary from scope to scope. So every scope, this is a transmission light microscope. It sends light through what you're looking at. And so when you look through the scope, you get a bright background. So I'm going to turn the light on. Light comes on down here. Up here is a lens. The lens takes the beam and tightens it, pulls it tight on what you're looking at. So whenever you use a microscope with what's called a condenser lens, really cheap ones don't have this. There is a control here that, I don't know if you can see this in here. It moves this up and down. And anytime you get on a microscope, and it's real easy to turn this knob thinking you're turning one of the other knobs. But if you get in a microscope and you look and this thing is like buried in the hole, it's not doing you any good. It's got to be up really close to what you're looking at. If it gets in just the wrong place though, it will send you a magnified image of the frosting on the light bulb. And so the background will get all speckly. And so if you have that, you just move the lens a little tiny bit so that it's focused on the light and not on the surface of the light, and that's fine. Okay. This is the first slide we're going to use. So if you can get in close enough to actually see the three threads. So this is the first thing you're going to do is draw the three threads the way they look without the microscope. Notice they're three different colors. If you don't have colored pencils, label each color. If you have colored pencils, feel free to put them in different colors, but make sure you label them, make sure it looks like this. Now I'm going to take this slide, which I have cleaned already. I cleaned it off ahead of time. You always clean your slides, otherwise you're looking at dust a lot. I'm going to take the slide just the way you looked at it and put it flat down on the scope. Open the little flippy thing. That is the technical term for it, the little flippy thing. You always put the slide in to the bracket as far back and in the corner as it will go, and gently close the flippy thing. Now some microscopes, because of the way this bracket works, when you put the slide on there, the bracket is way off in a corner or something. You still put the thing in the bracket all the way, and then there's little dials that you use to move the slide around. Now, underneath here is a little tab. If you look up here though, don't look at the tab so much, just look here. You always fumble for the tab, you feel for it. It opens an aperture inside that's called your iris diaphragm, and you can open it and you can close it. And I'll show you a little bit later what that does to what you're looking at. Now notice at this point that when I open it, it lights up those three threads. Just come back to me. One of the most important things you can do when you're going to work with a microscope, you clean your slide off. If you've got a cover slip, you clean the cover slip off. If the slide is already made with something on it, you take it and you hold it over a piece of paper, or you hold it up to the light and you look at it. Very often, I know you're looking through a microscope, but what you're going to look at is visible by eye. Like the three threads are visible by eye. So you look and you go, oh, it's up in the corner of the cover slip. Now I know where it is. I put the slide on here, I open up the iris diaphragm, and I move it until I see that thing I saw before light up. That puts it right underneath the lens. So you don't need to hunt for it. is that like I lit the threads up, I know the threads are gonna be where I'm looking when I look through the microscope. If you get a slide and this happens where you look and you go, I don't see anything, because what's on there is literally too microscopic to see, then the trick there is you move the slide until the edge of the cover slip lights up. You can see it through the microscope. And if you focus on the edge of the cover slip at low power, anything under the cover slip is in focus too. So now at least when you go hunting, because one of the frustrations of a microscope is that you're trying to find something and you're thinking, I don't even know if I got this in focus. Well, if you get the edge of the cover slip in focus, at low power, what's under there will also be in focus. And then you just scan back and forth looking to see if something will show up. Okay, so the powers are controlled here. This little bitty housing has got a 4-power lens in it. This, on our microscopes, is a 10-power lens. I don't know, some microscopes it's 15. But the total magnification is this times that. So I'm assuming it's 4 times 10, which would be 40. There's a little merry-go-round here. So there's a middle power that's 10. There's a high power that's 40. And there's an oil immersion, which we ignore because we don't use it in our class. Well, you might use it for one of the later labs, but we're gonna all hope that you don't. So you'll hear me call them low, middle, high, and oil immersion. So I've got this under here. The way to focus it, and I let the microscope be my glasses, so I always focus it with my glasses off. It's not necessary, you can focus it with your glasses on, doesn't really matter. I'm just used to doing it this way. You always focus, these are your focal knobs. There's a big one on the outside, that's the coarse adjustment. It moves this thing with a slide on it a lot. Once you get it pretty close to being in focus.
Here, you move the coarse one again so I can show the slide moving.
Okay. Oh no, the table, there we go. Yeah. Now, what I'm gonna do is I'm gonna look through the right side with my right eye and focus this until the thread gets pretty in focus. Is that there's an image of what it looks like. Make it a little brighter. Not entirely sure why this light is flickering. Now, there's something about this projector. What I'm looking at has a very white background. You want a white background. With a microscope, if this was what I was seeing through the microscope, I'd brighten my light up. But something about the way this projects, it makes the background yellow. That actually, not what you want when you look through a microscope, but you can see. And you can see the 3 threads. Now, I'm not done over here yet though, because I have 2 eyepieces. I've just focused the scope for my right eye. I want to focus the scope for my left eye, but I can't use the same control because that'll mess my right eye up. The left eye has, you can turn the bottom of the eyepiece. And so you always pick something really specific, like one of the little bitty threads in this case. So I can see the little bitty thread with my right eye, and now I can see the little bitty thread with my left eye. To tell you the truth, I'm assuming that that's seeing what I focus for my right eye, and that it's kind of ignoring the other side. But I don't know that for a fact. Oh, this is way too bright now. Okay, now we had the iris diaphragm, is I showed you before, it makes the spotlight. This is what happens when you open it and close it. You always feel it underneath for the tab. Okay. There it's closed. You can see it increases the contrast for a lot of these pieces. And there it is open. There we go. It kind of overwhelms the video system a little bit. But it makes the colors more obvious. So right now, this is set up for at low power. And we'll come back to this because question 2 is about this. But I'm going to show you some stuff. Don't come too close because you're going to need to look at that as well. This little merry-go-round, I'm going to go up to the next power. And we're barely looking at anything. It actually is in focus, but it's only in part focus. Here's what happens. Whatever you looked at low power, when you go another power up, you're only looking at a little bit of that because it had to make everything bigger. And so what we had in the middle of the screen was the space between the threads. That's pretty much what we have now. If I move the slide, there's the green thread, and there's the red thread, and there's the yellow thread. It looks brown under the microscope. But what that means is, if you've got a big space in the middle of what you're looking at, and you go up in power, all you're looking at is the space. Whenever you go up in power, take something that you can see, usually really well, move it into the middle, then go up in power and focus on that. For part 3, we're going to look at our own cheek cells. Cheek cells are very hard to see, but we almost always capture little bubbles of air under the cover slip of the cheek cells, and they have very dark edges to them. So I always tell people trying to find their cheek cells, as they go up in power, put a bubble of air in the middle, focus on that edge. Once the bubbles are in focus, the cheek cells are right there with them. Now you can look around and see the cheek cells. So the secret is not necessarily to focus on what you want to see, but focus on something you can see, and when you go up in power. So this is at middle power, and here's another thing about the way the microscopes work. At low power, there's what they call the depth of field. So if you imagine that I am a thread, when you got me in focus at low power, you could see the little bitty threads in me from the top of my head down to about my knees. About 3/4 of that thread was all in focus at once. That's the depth of field. Now that we're at middle power, we're only looking at about this much, which means some of these edges things and some of these threads are in focus, but the whole thread, which is pretty thick, is fuzzing out. At high power, you get a very narrow field of view. Is that on some things it's only 1 cell height. And so focusing at high power can be really, really tricky. I'm not even gonna try to focus this thing at high power, but I do want to talk about going to high power. So come on back in and look at this. For each one of these powers, now low power can focus a long way away from what you're looking at. Middle power, the lens is down in the bottom of this, it's got to get closer in order to focus. High power gets really close to what you're looking at. So when you use high power, you have to do a couple of things differently. Yes, you need to put stuff in the middle. You always need to do that. But when you move your lens, your high power lens into position, you don't keep looking through the scope. You lean away and watch to see if it's going to hit anything. Because some preps are pretty thick, and you can actually smush your cover slip with the housing here and break the cover slip. Theoretically, you could break the lens, but that pretty much never happens. So you watch that. Also, because it is so very, very close, you no longer turn this big old crank to focus, because it's too easy to drive the lens into the cover slip in the slide and break it. So you use the fine adjustment. The fine adjustment, this knob in the middle, it moves this, but you can't see that it moves this. And there's no way in the world I can get this thread in focus. And if you look, even the teeny tiny threads off to the side, this is in focus, but as soon as it curves away a little bit, it's out of focus. This one is out of focus until it curves into that narrow plane that's in focus. And then oil immersion is even worse. But unless, I will probably show you how to use oil immersion when we look at bacteria in a couple of labs. But other than that, nobody, and the people in the lab, if they're lucky, won't need to use oil immersion to see their bacteria. No dancing in the lab. Okay, back down to low power. You can stay on the screen. Okay. Question 2. Draw the threads the way they look under the microscope at low power. And again, label the colors or draw them with a colored pencil. And this leads into one of the tricky questions on the lab, which is, okay, the microscope has made this bigger. You can see stuff that you wouldn't see otherwise because it's bigger. Something else has changed between seeing it with your eyes and seeing it under the microscope. And I want you to tell me what that is. It is literally right here and right in your drawings. If you look at your two drawings, it should be an obvious difference. But it's weird that people look at the drawings and they don't see it. And that is the biggest clue I can give you, that is going to be the issue between what you see for question 1 and what you see for question 2. And question 3, what has changed that has nothing to do with magnification? Okay, I'm going to do these parts separately, so these videos are going to be relatively short. So we're going to stop this now.